everyone. I'm Steph, and that's Lisa together with Jenny. We're PD Connect. And today we have Dr. Giselle Petzinger, and Lisa's going to intro her for us.
All right. So Dr. Giselle Petzinger is an associate professor of neurology, a clinical scholar at the University of Southern California. She is a board certified neurologist specializing in the care of patients with PD. She divides her time between clinical care of patients and laboratory research. The primary focus of Dr. Petzinger's research is to better understand how to promote brain repair following injury and to translate these findings into new therapeutic treatments. That's cool. Dr. Petzinger has been involved in numerous federally funded research grants. I thought this was really cool. This morning, I went through some of her research. Of her numerous publications, topics include from enhancing neuroplasticity in the basal ganglia to understanding how exercise elevates dopamine receptors, to cardiorespiratory fitness's association to cognition, to perivascular space in types of PD, to personality traits and risk for PD, to context-dependent learning, to induced astrocytic changes, and so on and so on. Thank you, Dr. Petzinger, for being with us, and it's all yours.
Absolutely. Thanks for having me. Sorry for the technical difficulties. I apologize for that, and I am a little bummed because, well, anyway, I'll describe the videos because some of them are probably didn't transfer. But anyway, thank you again for inviting me. My name, as you know, is Dr. Petzinger. I was trained at Columbia University, so I'm a product of Stanley Fawn, who's a big Parkinson's guy, and love him to death. Anyway, so I've been at SC since about 2000, and what I'm going to do is walk you through some of the a lot of the research we've been doing for a while now in the field of Parkinson's, more specifically plasticity. What I'm hoping to touch upon is not only mechanisms, but also obviously something that's near and dear to my heart, which is how does exercise affect thinking? And we're going to get to that, some very new data that hasn't even been published yet that we're going to share today. First thing is to remember, so it takes a village. I've worked with a lot of different people, most everyone, 99% at USC, some people in San Diego, some people at University of Virginia, but I work with a lot of very talented people at FC. We don't have a good football team, though, I can admit that. So any of those watching Cal game yesterday, we were killed, I know. So why do we think about plasticity in Parkinson's anyway? Well, this is just a cartoon that was published in one of our reviews. And what you're seeing here is kind of an outline of a brain, and it's kind of a light blue shade. And you're seeing, just as an example, these yellow arrows that are connecting up. And all I'm just trying to show here is that there are circuits in the brain. There are parts of the brain that connect up with other parts of the brain. And these connections, which we call circuits, are really what drive behavior. And a lot of what we've been interested in understanding is there evidence that we can influence these connections? And the long story short is, yes, we can. And I'll show you some of the data that shows this. Okay, so just to remind ourselves, you know, we've been talking about neuroplasticity for a long time. People have heard this term. This term came out of Decade of the Brain in the 90s. And in the Decade of the Brain, you know, people began to recognize or really revisit the idea that the brain changes throughout our life, and even as an adult, it changes. And it's not just about new cells. You've heard about newborn cells, but more importantly, it's the idea that there is reorganization within the brain that's happening throughout our life. And that reorganization happens at the level of connection. So that's the first point. Plasticity is really about this reorganization of these connections within the brain and through our life, and then that it never stops. The second point about understanding what do we mean by neuroplasticity is that when we talk about this reorganization, we're really talking about it at the level of the physical connection. And that's called a synapse. So we're going to be hearing that term over and over again, the synapse, which is where the connections are actually happening. And as you can imagine, there's a physical component of it, meaning a physical touching of a connection that we're interested in, obviously. But there's also what's called a functional aspect of it, which means that the connection's there, but the way it's interacting with one another is changing. So there's a functional and physical changes that are happening within synapses. And that's really, again, what we're talking about with plasticity, how those changes happen. I think the other idea is there's these bigger ideas of plasticity in terms of what we call homeostasis. And the word homeostasis is really reaching a type of balance, right? Or what we call new normal. And homeostatic doesn't necessarily mean returning to something, right? So let's say, I'm born and obviously my brain is changing, I'm getting older. When we think about homeostasis, it's not going back to infancy, okay? What homeostasis means is creating a new normal. So change doesn't mean going back. Change just means different, and homeostasis is the same idea. The brain might find a way to kind of fix itself or make alterations, but it doesn't mean it's going back to what it was. It could be completely different, in fact. And we need to think about that because there's always kind of this idea to think that, oh, if I repair something, that means it's got to look the way it was 10 years ago. That's not necessarily the case. These changes that could happen could be different than what it was, but it's still giving me functional benefit. And that's really important in the field of plasticity because we recognize that some of the things we're seeing that are different aren't necessarily the same way it was. And I'll explain that a little bit more as we go on. Taking it back to what does it mean for patients? What does it mean for me as a person? Well, the idea is that at the end of the day, the reason we're interested in plasticity and change is that we believe, number one, it's really the way that the brain encodes learning. It's a way for the brain to get better at something, right? So when I'm making changes to these synapses, I'm not just doing it to do it. I'm doing it because it's encoding information there, and it's encoding useful information. And we got really excited about that a number of years ago, because it really was getting at the idea that I could maybe influence this learning. I could drive this learning in a way that's helpful, that may be beneficial to a patient. And of course, when we think about that type of learning, we're talking about rehab, right? The idea that I can get you better by practicing and doing something. So I'm encoding some behaviors that are helpful. Does that make sense? So again, you get this feeling of plasticity at multiple levels, at a very molecular level. What am I talking about when it talks about the brain? At a systems level, what's happening in terms of what's changing and how they're related to each other, this new idea of homeostasis, the brain is trying to reach a new normal. And then finally, it's the principle of how we learn. It's the principle of how we encode information is what plasticity really is. And we can borrow that in the rehab field. Okay, so interesting. Okay, so I'm just showing this to remind ourselves of all the amount of work that's been done in the exercise field for at least 20 years now. And really the take-home message has been that practicing motor function turns out to be good and helpful and powerful. And there's a lot of ways to do that, right? There's not one way to get better at gait and balance. As long as I'm practicing gait and balance, I should, in theory, get better at gait and balance. That's called learning, guys. If I study math, I'm going to get better at math. If I study French, okay, maybe I'll learn French, but the chances of me doing better in algebra is pretty slim. What does that mean? I mean, there's some specificity to these effects, meaning, yeah, gait and balance, there's a lot of gait and balance in all these things. I'm going to get better. So I think the other take-home point is there's no magic exercise. There's no the exercise. It's what you love to do. As long as I have gait and balance, that's great. It just so happens some of the original work that we did happened to be in treadmill. Why? I don't know, because it influences dynamic balance and gait. Voila, no surprise. And of course, in treadmill, we can get a lot of practice. So the original work that was done, which focused on treadmill, was just honing in on the idea that if I get a lot of practice, I'm going to get better. And as I said, since then, a number of different studies using a number of different types of exercise, even dance, has showed improvement in motor features with gait and balance and being particularly improved. And I apologize because this will not work. Oh, yes, it does. There we go. So this was our original work. So some of our original work in gait and balance, this is now over 15, 18 years now since we've done this original work. This is one of my patients before exercise gait and balance study. This was using amplitude training with Claire McLean. And this is after. So that was some of the original work that came out with our group here at USC. So we were able to show that if you practice gait and balance, you get better. Wow, right? Okay. The other thing that was important to keep in mind was when we were doing these initial studies, there was no rehab in Parkinson's disease. There was none. Why? Because people didn't think it was relevant. It was all about drug therapy. No one thought that exercise could be helpful at all because no one thought that plasticity could happen. So we were the first group to show that people were wrong. No, in fact, you could get evidence of plasticity. And that work came out originally in animal work that we did here, again, back in 2004. So just to show you some of that original work. So this is humor from my husband, who is also my collaborator. This is translational work. So we were doing all this work on treadmill, and then we moved it to the mouse. Obviously, we can look at mouse brains. And one of the first things that we asked, let me see if this works. Yes, it does. So this was the original work that we did in the treadmills. So these are Parkinsonian mice that we put on treadmills. And basically we ran them for 28 days. And what we ended up doing was we ended up increasing the speeds, very similar to the human, which is we kept driving them harder and faster and harder and faster, really to see, first of all, how well they could do on motor tasks afterwards. And what we were able to show, in essence, was that, here we go. Oh, this one doesn't show. Well, basically, this tape here is just showing two mice on a treadmill. This is a Parkinsonian mice that was been practicing, and this is a couch potato mouse. What we were able to show was, in fact, that the animals who ran regularly on a treadmill did better over time. No question about that. And so we were able to show that not just on a treadmill, but we were able to show that on a rotating rod, accelerated road rod. So here was the exercise group. This is a no exercise group. And we showed over time that the animals that ran, they were both Parkinsonian, no question about it. But the animals that were practicing running, not only could run better at the end, they were running amazingly better, but they could also do better on other motor tasks. Now that sounds silly, doesn't it? Of course that's obvious. But the time that we published this in 2004, this was the first time this has ever been shown. No one had shown this before, right? That we could get improvement while an animal had Parkinson's. The papers that had been published before that, and there were a few, they looked at it as protection. In other words, they try to show that exercise protected from toxin injury. But ours was the first paper to show that now an animal that clearly has Parkinson's, do they get better? And the answer was, yes, they do. So we saw mode improvement. We then began to take and look at the brain because we were interested in understanding what would it do to the brain, and particularly what would it do to, hold on one second, what would it do to dopamine levels? So let's look at dopamine levels, right? This is looking at dopamine levels in the brain of an animal. Now, you're going to see two groups of animals. They're both Parkinsonian. But in one group, this is the exercise group. This guy was rocking it. This guy was doing amazing at the end of 20 days. He was amazing. He looked great. He was on a rotor rod. He could do all these motor tasks. And the animal that wasn't doing well, who wasn't exercising, was falling and not poor balance and everything. So we took out the brains and we looked at dopamine levels. Now, I want you to tell me if there's any difference in the size of these two columns. The answer is no. So there is no difference in dopamine levels. This data has been, anyway, this data is old data. This is 2007. We clearly showed that there was no difference in dopamine levels. And basically, people have misquoted us in so many different ways, I can't even tell you. But the bottom line is there is no difference in dopamine levels. What does that tell you? I'm telling you that this improvement in behavior is downstream. It's in the circuits. It's not changing dopamine amount, all right? In fact, it didn't even change cell count. We published this back in 2007. There is no difference in cell number. There's no difference in dopamine levels, right? So what is going on? If I see a change in behavior, it's not from either one of those. And in fact, we looked at neurogenesis. There are no newborn cells. We haven't bothered to publish that, but there is no evidence of neurogenesis, not in at least the animal models, and no differences in dopamine levels. So what is explaining this effect? So we looked at, pardon me, we did see one thing though, pardon me, let me back up. We saw that there were changes in the amount of dopamine release. So let's say if I could just grind up the whole brain and look at total dopamine levels, I can tell you there's no difference. But if I can measure, I can stimulate cells and see how much dopamine is being released. What I can see is that dopamine is being released faster. So all I'm showing you is a slice of a brain that says 1, 2, 3, 4, that's the basal ganglia. And down below in red, in a big circle, you see these columns in light gray that are higher than the columns in dark gray. Those are the exercise, non-exercise animals. And what I'm showing you there is that dopamine is being released faster. So what exercise does is it doesn't change the amount or the cell number, but it does change the availability of dopamine. So it releases faster is what I want to say. It makes it more efficient. Okay. One other thing it does is it changes the dopamine receptors. And so this again is old. This paper came out in 2007, I believe. And I'm just showing you that we measure dopamine receptors by transcript, by protein. We measured in a lot of different ways within the brain of the animal. And the reason we care about the dopamine receptor, particularly type 2, is that in Parkinson's, the receptor drops out. The receptor decreases in amount. And that receptor is particularly important for motor control, for motor movement. So we saw that it went up with transcript, which is gene expression. We saw it went up with protein levels. So we decided to image it with a PET scan. And so we worked with somebody that made a ligand, a radioactive ligand that was colorimetric that we could image. And I could image to see what was happening to the receptor itself within the basal ganglia. So I'm just showing you, this actually made the cover of Movement Disorders in 2010. Basically, what we're showing is on the left is a normal animal. What you see in red is the amount of dopamine receptor type 2. What you see in the middle is a Parkinsonian animal. You see it drop out there, right? So the dopamine receptors start dropping out. And then what you see to the right of that, it says MPTP plus exercise. That's the Parkinsonian animal that was exercising. What you see is the receptor number coming back in orange. That's just pure exercise. So exercise actually changes the amount of dopamine receptors, okay? There's been a person in British Columbia who replicated this work that we did. We actually did this again in humans. The work in humans came out the next year. So this is the work we did in humans that showed the same thing. So what I'm showing you here is basically this picture down below. I apologize, it's small at the bottom, it's in red. This is showing you the increase in D2 receptors. This is a human brain. This is a patient that, and this is looking at, we had 8 patients, but basically what we did was we put them on the treadmill. We imaged them before and after the treadmill exercise, and we were able to show in red an increase in D2 receptors. That was the original work that showed it, that work, our work at USC, that we had an increase in D2 receptors in mice, and we are able to replicate that in humans. And then maybe 3 years ago, they did the same study again that we did using something similar to show an increase in D2 receptors. So what am I saying? What I'm saying is that with regards to dopamine, what we have evidence for so far, and this has not changed since our original work, we see no difference in cell number. We see that dopamine is more efficient. The total amount hasn't changed, but it's released better. And we do see a change in a receptor, a dopamine receptor that happens to be pretty important for mobility. So that alone may be part of the story for why people get better. But we decided to go on and look for other things that might be changing. And those other things included things like another neurotransmitter called glutamate. Now, this is a combination of a number of papers, of 4 papers that came out on glutamate. Why do we care about glutamate? Remember I told you about how receptors have function besides, and synapses have function? In other words, it's not only about the physical connections, but it's also how they're interacting. So we started measuring glutamate because it's another very important neurotransmitter in Parkinson's. And long story short, there were about 3 or 4 key features we saw in glutamate that had been changed with exercise that we thought were beneficial. Some of them, like I'm just showing you cartoons here, were the type of glutamate receptors we saw change that seemed to be more efficient. We saw changes in the amount of glutamate. There was a buildup, and we saw it go down with exercise, which was healthier. And then finally, we also saw a change in the type of glutamate receptors that were better for conducting energy. So this was all came out in a couple papers from 2008 to 2010, all showing evidence in the animal brain that not only did we see changes in dopamine, but we saw changes in glutamate as well that we thought were helpful. Okay, so that's the functional aspect. These are just the chemistry of the brain that's changed with exercise. Now let's talk about the physical connections. Remember I told you about synaptogenesis. This is a physical connection, guys. This is what a physical connection looks like. It's literally one cell connecting with another, and there you see a synapse. That's what that is in cartoon form, a synapse. And the reason, remember, we care is, remember, these circuits for behavior are made up of these things. So the question is, what happens typically in Parkinson's when the dopamine levels are dropping is these synapses are actually physically falling apart. It's not like you lose all of them, but they are disconnecting to some degree, and that explains why people get slow. The loss of connections is a big part of why behavior changes in Parkinson's disease. So what happens with exercise in animals? Well, basically the first thing we did was we looked at the cells within the basal ganglia and we outlined them. These are all kind of branches of a cell. This is how it connects up with others. It sends out branches. So they have all these branches. And the first thing we counted was just the number of branches of a cell to see if that changed. This is examples of 3 cells. This is a cell from an animal that's normal. This is a cell from an animal with Parkinson's. See the difference, guys? Look at all the loss of branching there. And this is a cell in an animal with Parkinson's exercise. Look at that. The difference between sailing and a Parkinson's, and there's a Parkinson's animal. Look at how much more branching there is. So besides changing chemistry, changing dopamine, changing glutamate, we actually see physical changes in the amount of branches that are happening. And that's just an outline example of how we outline cells in the brain that we can trace them and see how extensive they are. So the bottom line is exercise obviously changes branches. So how about the physical connections of these cells, and what does that look like? Pardon me for a second here. Just trying to. So I'm just showing you, just to remind you, I was showing you these branches. See the branches? There's a cell in the center. There's all the branches. I'm just showing you examples of what branches look like. And then when we blow up those branches, those are the little physical connections that should be there. When you have dopamine loss, you can see they're stripped. There's a lot less of them. So there's less branching and less connections. And so what happens with exercise? So what I'm showing you is a work that was done by one graduate student in the lab who spent a lot of time counting connections. And what he would do is he'd fill the cells, and then he would see all the little connections. And what you're seeing in C, panel C, you see all those little dots coming off? Those are all the synapses, right? They're physically filled with this dye, and he's literally counting them all up. Look at the difference between sailing and MPTP below that. The MPTP below that is the Parkinsonian animal. So I already showed you there's less branching, right? There's also less of these synapses. You can see that. And what we see to the right of that, it says MPTP plus exercise. That's the Parkinsonian animal again that was exercising. And what we see again is not only more branching, but more connections. So far then, again, where's the evidence of plasticity? Well, in the animal, certainly, and this is a Parkinsonian animal, we have a lot of evidence for that. There's no question about it. We have dopamine changes, we have glutamate changes, and we have not only branching, but we have evidence, not of neurogenesis, but we have evidence, clear evidence of synaptogenesis. Okay, so that was a quick run through about 10 years of work with like 100 people helping out with this. It's a hell of a lot of work, but basically trying to take home the point. Yeah, there's evidence for plasticity, okay? Done. The question is, now we're also asking how and why is this happening? What's the mechanism of it? before we go there, one of the questions that we're also, at the same time, we started asking about the different types of exercise. Does it matter? And that came up because as we were doing all this work with skill-based exercise, which is really focused on gait and balance, our aging colleagues were doing a lot of stuff with aerobic. And aerobic means getting your heart rate up, but it doesn't necessarily mean gait and balance practice, right? So skill-based means you're doing things that are more goal and task-based, where I'm getting better at function. I'm getting better at doing things, for example. But aerobic exercise is not that at all. Aerobic exercise has nothing to do with, am I gaining a goal or learning a skill? It's just the question, can I get my heart rate up so that I'm extracting oxygen better? So that's really aerobic exercise. And again, we got interested in that because of two things. One, because the aging field was looking at aerobic exercise, we wanted to know if there was a difference between what they were doing or what we were doing. And number two, I got very interested in cognition and how we can help thinking. Because remember, a lot of what we've been focused up to this point really had been on gait and balance. But as you and I know, Parkinson's can absolutely affect thinking as well. And there were still a lot of questions that were, remained about, well, okay, can I expect to feel, that my thinking has improved? Is there any data for that? This is just reiterating what the difference is between skill and aerobic. And I'm going to just show you some work that was done by a colleague of ours here at USC. And he tried to take this question on in a rodent model. So this is another Parkinson's model. But in this case, I'm going to show you, how do we do this sort of study in an animal? Well, one way to do that is, if you look at these two wheels, you can see there's a difference between them. And one of the biggest difference is that one wheel is complex. There's actually some of the spokes removed, right? And so that's going to be quite challenging for any animal to get on that, because they're going to have to pay attention to those spokes versus a smooth, right? Both these wheels are put on mechanized rolling wheels, so rods that are rotating. So these wheels are put on those and they're moving, and the animal has to stay up. So they're not moving this. There's a mechanical rod underneath that's spinning slowly, and they have to keep up. So that velocity is increased every time the animal is showing some improvement that they can do it. We make it harder. We go up on the speed. And the question became, is there a difference and this question we're looking at is blood flow. Yes, there's a question. Someone put a hand up, or no? A hand up? No? Okay, so what we can do, this is just a quick outline of the study. We can put a dye in the animal that's going to trace blood, that's going to help us tag blood. And why we want to do that is we're going to want to see where the blood is going in the brain. In other words, if I've been exercising an animal with aerobic versus skill, and I have them do it for about 8 weeks, then I look to see, does the blood flow distribution matter? Can I actually change blood flow distribution? Because they're both exercising, right? But can the blood flow distribution change simply by what I'm doing? And the short answer to that is yes, it can. So here you have the difference between skilled exercise and non-skilled or aerobic exercise. And what you're looking at red is the blood flow. And remember, the blood flow is tagged so that I can see it and put it on film. And basically what you see to the left is an animal that's been working out on the skilled wheel versus ones that are more aerobic. And in this case, the areas of the brain that we're seeing with the skilled is we're seeing areas of attention, and we're seeing areas, PFC, prefrontal cortex is what that stands for, PFC, prefrontal cortex. So we also see it in aerobic, don't get me wrong, but we see it particularly heavy in the skilled. And the idea, which is kind of cool, right? Because remember, they're both exercising, right? So the other cool thing about this is it totally suggests what we love. And that is, the brain is totally an active participant here. It is not passive at all. It's helping making these changes happen. I mean, the reason this changes is because the brain has made a decision about its metabolic need. It's saying, I am working out in a skilled process. I need to be digging down hard on what I'm doing. And it's putting this incredible metabolic demand on these certain circuits, and blood responds. Isn't that amazing? So again, they're both exercising. The difference is how I'm using my brain. Isn't that amazing? So I can totally change. Again, exercise is not a passive event when it comes to the brain. The brain is very much active in that. Both are gaining benefits, but the distribution seems to be different. Excellent. So let's start asking the questions. Well, so what does it mean in terms of humans? And I apologize because we've done a couple studies, and this one is sort of just coming out, and we're working on this paper right now. But this is a study that we're looking at trying to address this question in humans. And there have been a couple groups that have been trying to address this question after we put out this paper and started asking it. Then it became very popular. But basically the question became, does it matter what I do in terms of cognition? And one of the ways that we addressed it was we began to look at aspects of, again, skill-based exercise versus cardiovascular-based exercise. And we collected information from people that we thought was related to skilled exercise. So hear me out for a second. we collected something called motor fitness. We collected a motor fitness parameter. In other words, if I wanted to know how much skilled exercise you were doing, what I would do is I'd put you through a gamut of different little trials, and I would collect that information, add those numbers together, and say, this is your motor fitness score. And similarly, if I wanted to know how hard you've been working out aerobically, I collect a cardiovascular fitness score. And there's a way to collect cardiovascular fitness, which is looking at how well you extract oxygen. So we collected a motor fitness score by taking a number of these different batteries, the PPT, timed up and go, 10-step test, and short physical performance battery. And we created one composite, basically a kind of a type of composite score for that. And then for the cardiovascular score, we collect something called VO2 expenditure. So those are two things I can now, every patient of mine who's been doing exercise, I collect that, and I collect the cardiovascular fitness score, and I relate that to cognition. All right, so this is the data that we have. If I look at motor fitness, how motor fit you are, and that means how agile you are, how well you're able to move through these different tasks, what I can see is that we see clear associations with several cognitive domains. Here's attention, like we saw in the animal, executive functioning, language, and memory. So we see a very strong, this is N of 100 patients. So it's not a huge study, but so it's about 100 patients. But we see these 4 domains being affected with that motor fitness parameter. With cardiovascular fitness parameter, we see below, we also see executive function. So it wasn't like we didn't see any, but interesting, episodic memory. And what was kind of cool was visual spatial memory. So what I'm saying is both of them have benefits on cognition, whether you're doing aerobic or whether you're doing skill, but there may be some slight differences. And we're still exploring this. But the hint is that there could be some slight differences in terms of the type of benefit I get. And that's not too surprising. And as I said, it could be, as an example, that skill-based may give us a little bit better gains in attention, but there's also a lot of different domains of cognition where you get benefits, whether I'm doing cardiovascular or skill. Now, this is what we're talking about is thinking parameters, okay? All right. And the paper that came out last year or the year before basically showed that, as an example of cardiovascular fitness, that we saw improvements in the size of the brain in the area of some deep structures called the thalamus and pallidum. So people who, in this case, were more cardiovascularly fit, not only do we see improvements in cognition, but we saw certain structures of the brain were bigger in patients as well. The work that's being done on the motor fitness is still, we're not quite finished with that. Hopefully we'll have a paper out in the next year showing what parts of the brain seem to be particularly effective with motor fitness. So that data is coming together right now. But good message, not only, again, we see benefits in both types of workouts for cognition, but they may be slightly different, particularly with motor fitness, maybe tapping in on more attention-related aspects, but overlap in both. We see EF in both, and we see, for example, memory in both. Aerobics seem to have a little bit more visual spatial, which I have to say was surprising. I thought that would be more skill-based, but okay. So finally, another study that is coming out where this paper is hopefully should be in press and within the next few months. And we asked another question, because this question also comes up. It's very popular right now in the field. And this question now is about intensity, right? The question of how hard should I be working out? And the reason we care is because the question of physical activity intensity is an old question when it comes to Parkinson's disease. And how old is it? Well, we're talking the 1990s for sure, where some of the original work in Parkinson's disease showed that if you were more physically active, my risk for Parkinson's was low, right? You guys remember that? So the whole field of physical activity was an epidemiological question that came out in the 1990s saying, can I reduce my risk of Parkinson's? And the answer was yes, you can. People who were much more physically active, particularly intense, had a lower risk of Parkinson's disease. But the question we asked, we turned it around and asked, okay, but now I have Parkinson's. Does it still matter? Should I still be intense? And of course, the question that I was interested in was back to cognition. And again, why do we care about cognition? Why do I care about thinking so much? Well, because we don't have any good drugs. That's the bottom line. We have no good compounds. In fact, there's no good compounds for any field of thinking, whether it's traumatic brain injury, whether it's Alzheimer's, whether it's Parkinson's, whether it's aging. We have no drugs. I mean, those ones that they market are honestly a joke. They don't do very much at all. So therefore, lifestyle is important to know. And the aging field has totally taken this on, right? There's such great papers in aging that have clearly showed some really nice data, very convincing data. You want to find a great paper, 2004, the FINE study in 2004, published by the Netherlands, looked at aging. And they totally showed that if you wanted to have effect on cognition, you better bring up your act. You got to bring it up intense. So the question is that true for Parkinson's? Well, So we were just finishing a study. This is part of a Department of Defense study. It's a longitudinal study. We're following people for 2 years. By the way, the FINE study followed people for 10 years, starting at age 70 to age 80. That was a great study. So we don't have people for quite that long, but this is the beginning of the story. So the first thing we just asked right out of the gate, how does intensity relate to how I'm doing now? So I have 100 people. I'm monitoring how active they are. This is an observational study. I'm not doing a darn thing. All I'm doing is telling them, do what you normally do. This is in Southern California. There's a lot of different things people do. I said, I'm going to give you a wearable. You're going to wear this for me. So people wore these wearables. They've been doing that every 3 months for the last 2 years. They use these wearables and then they send it back to me. And I take that data and I tell them how hard they're working. And so what we can do is we can look at moderate to vigorous intensity, how much they're spending time in moderate to vigorous intensity and how much they're spending time in light. So let me explain, what does intensity mean in the context of physical activity? Well, what people really mean is metabolic expenditure. How much energy am I expending? When people talk about physical activity, they're talking about energy expenditure. And usually that they compare it to how much energy you're expending when you're just sitting, when you're sedentary. So that is called, let's say, 1 metabolic expenditure. If you are working very hard, you can get up to 3 times the amount. So 3 metabolic expenditures. Anything less than that is considered light. So that would be maybe a very light walk would be something between sedentary and 3. But anything more than 3, moderate to vigorous, you're working out pretty hard enough that it may be challenging to carry on a regular conversation. Okay, that's how hard moderate to vigorous is. Okay, so at the same time, as a side, we ask people to just write down how many times they're working out a week, how many minutes, how often, and what different types of exercises they were doing. That was on an exercise log. So on one hand, people were using a wearable device. This is something that they was wearing around their waist, and it was an actigraph. So it captured how hard they were moving. And at the same time, they were writing down, oh, I do this many minutes. I went to the gym and and then we captured frequency and we captured variety, all on a log. The take home point that we have is this: out of the gate, Mod Vig is rocking it. Moderate to Vig, and I'll show you this in a second. Moderate to Vig in every single domain is showing benefit if you're in moderate to vigorous exercise. And guess what? Duration, frequency, and variety, nothing. Not a thing. It doesn't matter with frequency, doesn't matter with variety, doesn't matter with duration. In other words, just recording how many minutes they're working doesn't mean anything. And I know that sounds confusing, but I'm going to just explain it in this way, in a way. The truth is, we kind of know this in that one would say, look, it's not about the time, it's about what you're doing, right? It's about, so what are you doing during this time? How hard are you working during this time? It's not just about doing something, it's about what you're doing. How hard are you working? That's what matters more than the minutes you say you're doing it. And I would say the same thing is true for anything that we do, right? Let's say I'm studying for a test. I tell, my friend, oh, I've been out, studying for 3 hours. Okay, but how much time were you spending doing practice exams? 15 minutes. Okay, well, the practice exam is really going to be the difference here. Does that make sense? So it's not so much about the minutes or even the frequency. It's about the quality. What are you doing during that time? And that at least seems to be suggestive of what we're seeing so far for cognition. And again, we didn't see anything with light. It's all with Mod Vig. So moderate to vigorous, visual, spatial, memory, executive function, and global with moderate to vigorous intensity. And this is continuous minutes, meaning the more minutes you spend, the better. And this is not in bouts. So people are just capturing minutes any way they can. This is not necessarily in 10-minute intervals. So the nice point about that is, yeah, the WHO has no recommendation that you have to do this in 10 minutes. They threw that out 3 years ago, 4 years ago. All they say is spend time moderate vig. They don't say anything about bouts anymore. And our data supports that. Doesn't have to be in a bout at all. Can be any minutes. As long as you're collecting minutes, it can be, short or long. It doesn't really matter. And that's so important because I feel like that's a lot about real world practice, isn't it? Now, obviously, when I do moderate vig, I could get a lot of that, right? I get a lot of brownie points if I do that. It's nice. I could do an exercise. That's a way for me to set time aside to say, I'm going to do mod vig. But the other take home point is the real world practice of this. That means anything I do that's mod vig. If I have to make a choice between an elevator or stairs, and I could get mod vig out of climbing that for a few minutes, that's going to count. Of course it's going to count because the brain doesn't know that, you only did this 3 minutes. It doesn't care is what we're learning. as long as you do mod vig. Now, I'm not saying that, again, exercise is great because it's structured. I can do this together with other people and I can do it at one time, but it's also reminding us that everything I do during the day is going to count for this. So whatever I do during the day, as long as I try to spend some time keeping active, non-sedentary, that's always going to be good. People may wonder about the sedentary data. We didn't find any bad things with sedentary data, at least for cognition. A lot of the sedentary work in general historically has mostly looked at mortality. There's less looked at with cognition. If you want to look at that, that would be in the aging field. Most of that, again, is mortality. There's some data for cognition, but mostly there isn't so much. Yeah, there's some for sedentary cognition. The bottom line is, in our Parkinson's study so far, we had sedentary behavior. It was actually a little harder to capture for us, but we didn't seem to get any negative things, which is ironic. You would think we might, but we don't. That may be the design of the study, but we definitely saw stuff with moderate vig. We're taking this study to the next level because remember I told you I've been following these patients for about 2 years. So we're about to ask the prediction question, because in the FIND study, the aging study published in 2004, not only did they show that it was intensity more than duration at baseline, in other words, how I did at that time in aging, intensity was better predicting how I did than duration even. So that's already been shown in aging, but it also predicted how these older adults did 10 years down the line. In the aging field, 10 years down the line, they showed that patients did not get worse and sometimes got better. So that is, and again, we don't have that data in Parkinson's. We're about to finish collecting that data in the next 6 months and hopefully have an answer for you. But so far, we can totally say that mod vig matters. We also looked at the 150-minute time point. So if you look at the WHO recommendations for physical activity, I'm telling you, this is already out there. Okay, we're this is not new. It just hasn't been looked at in Parkinson's disease. In the aging field, basically says adults, everyone should be out there working hard. Everyone should be out there moderate to vigorous. And the recommendation is 150 minutes per week. So do the math. It's about 21 minutes a day on average, right? Now, again, you could clump that all in one day if you like. You could do a whole day of this exercise session and you're done, right? Or you could do 21 minutes a day. Now, 150 is great. 200 minutes is even better, right? But all we're going to talk about is 150 minutes. When we took those patients and split them by those people who were hitting more than 150 minutes versus those that were not, we saw a couple things stand out. And those things were particularly true in executive function and visual spatial. So we do see benefits across the board, no question about it. But those people that are hitting about 150 minutes a week over, versus those that were not, we're seeing gains, particularly in executive function and visual spatial, which is pretty cool. And then this is a very complicated picture. This is, again, the paper that's going to be coming out. Basically, we're looking at connections within the whole brain. And it's a pretty colorimetric map looking at frontal regions, temporal regions, parietal regions, occipital regions. Take-home point so far, what we're seeing is frontal regions are stronger. These are people that are hitting 150 minutes versus those that are not. Frontal regions are very strong. Hippocampus, which is memory, is very strong, and brainstem is very strong right now that we're seeing. Okay, how are we doing with time anyway? What's the time, guys? Are we?
It's like 3:24. You're fine. We're great.
Okay, so all right. So then the next question is, so what's happening? I mean, we talked about how exercise is improving connections, how it's improving neurotransmitters. One of the things we became interested in part because of physical activity and the idea of metabolic expenditure was to look at metabolism. So one thing is we know that exercise, at least in the periphery and the muscles, exercise can change metabolism, right? And the question is, can exercise change fuel or metabolism in the brain? And that's a pretty cool question. So we began to explore that. And one of the reasons we also felt pretty confident that we should look at it had to do with this transcript. And I apologize, a very, very big slide. But again, this paper was published about 3 years ago, but this is looking at this factor. It's called hypoxia-inducible factor. It's a gene. It's a gene that turns on other genes. And we basically took a hunch that this gene could be upregulated with exercise in the brain. And the bottom line is, this is just showing that, anyway, we saw this increase in this gene was going up and went up by 5 days of exercise in the animal. Why we're so interested in that is because it regulates metabolism. So it's a gene that regulates metabolism, and it was skyrocketing in the brain of mice. And we thought, wow, we got to look at this. And of course, with physical activity, the idea that physical activity was improving cognitive function, we said, okay, let's look at metabolism. So one of the things that we looked at, though, in terms of what this gene turns on, is it turns on, and I'm going to explain this picture in a second. It turns on a metabolite called lactate. It changes the metabolism of lactate. Now, don't confuse it with lactose, which is milk. This is not milk. And don't confuse it with lactic acid, which would be very painful in the muscles, but is perfectly fine in your gut and is in fermented foods. So this is lactate. So lactate is something that is produced in the muscle, and it turns out is actually a fuel source for the brain. Most people think about glucose as being a fuel source, but it turns out that lactate is also a very important fuel source. Now, why am I showing this picture? Because I'm not showing lactate. In fact, I'm showing something very different. I'm showing a cell, and I'm showing a cell that's blue. And what's kind of interesting about this cell is it has little blue end feet onto that red structure, which is a blood vessel. And what I'm trying to say here is there's a cell called astrocytes that basically attach to a blood vessel, and the other part of them attaches to, you should recognize that in cartoon form. I could give a quiz, but I won't. That's a synapse there, right there. Okay, so guess what? Look at what that astrocyte is doing. It's connecting blood. So this is blood that's coming in from the periphery. Remember, blood comes in from the periphery. It's connecting a blood vessel to a synapse, which is a brain connection. Isn't that cool? So that astrocyte is like the best thing in the world right now, right? Because it's one of the ways that fuel gets into the brain is through astrocytes. Astrocytes have all these really cool ways of moving fuel sources that are coming in from the periphery, from things I eat or the lactate that I build up, and it moves it, it shuttles it all the way to where it's needed, which is connections. Because you and I know that if I'm thinking real hard, I need something to support that connection, right? I need something that's going to provide enough fuel so that connection has enough energy support to work hard, right? It needs fuel like anything else. So we said astrocytes are what we need to look at. And if people follow the field of Parkinson's right now, astrocytes are like the hottest thing right now. They have so many interesting functions, including immune functions, by the way. They play a role in inflammation. But more obviously, they are a key component to metabolites. No question about that. No one would argue that. So we started looking at astrocytes, going, okay, physical activity is good, exercise is good, astrocytes, maybe they're changing. And lo and behold, they are. So here we go. These are literally, now obviously I'm just showing you single cells. Again, this was a hell of a lot of work by a graduate student in the lab who basically took animals and ran them and looked at astrocytes in different areas of the brain. The prefrontal cortex is executive. This is I was showing you that area. There was a lot of blood flow there when I was working really hard in that skilled thing, right? The striatum is the basal ganglia, which is an area of the brain that we all know is involved in Parkinson's. He looked at these two areas and just said, hey, let me exercise them, and I'm going to take a brain out every week and just see what's happening to these astrocytes with exercise. And what he showed is that these astrocytes are getting huge. And look at all the branching. These guys are going nuts. They're reaching out to all these different synapses, trying to connect up. Isn't that great? So it's collecting fuel and it's moving it to a brain connection. So that was pretty cool. We were pretty happy about that. But he also got very interested in looking at lactate itself. And so this work, what he did was he took those astrocytes out of the brain and he put lactic acid right on those little astrocytes. And one of the questions he asked is, what else can these cells do? They're so amazing. Obviously, they create fuel sources. They move fuel around. That's awesome enough, but that's just not enough. These guys are on fire. He pours lactate on them on a little culture dish, and he measures, what are you guys making? And he finds out, look to the right. So the white is astrocytes without any lactate on them. And in red is what these cells are making when lactate is being delivered. And all those little letters down below are all sorts of different neurotrophic factors. These are like fertilizer to the brain. Neurotrophic factors are like super duper fertilizer for the brain. They help connections form. And lo and behold, not only does he see the astrocytes getting fatter, but he can show that if I put lactate on these astrocytes, they're going to start spewing out trophic factor. So these astrocytes are doing amazing things in the context of lactate. So what's the scenario you imagine? You imagine that I'm working out, doing exercise, and I'm getting lactate building up in my muscle. It's being carried by my blood because my heart's pumping hard. It's making it to my brain. And in my brain, it's providing a rich fuel source for connections in my brain. And it's probably also facilitating fertilizer, trophic factors as well. So all the things that we think of that are healthy and important for keeping my brain healthy, astrocytes are one heck of a source for those good things that the cells that exercise can do. So he also, remember, he took the cells out of the brain, but in this case, he also injected he actually injected the animal with lactate, so like in the muscle, and he was working out, so that lactate is moving to the brain. And again, he looked at neurotrophic, he took the brain out, measured neurotrophic factors to show that it was increased. So not only do we see it in the cell culture, we actually see it in an animal. We can measure the neurotrophic factors going up. So that was a paper that came out about 2 years ago. And that's all I have to show today, guys. Just to remind you, it takes a village, and my village is pretty darn big. And again, people at USC, Michael Jakowick is my partner in crime. He's a PhD. He's also runs the basic science lab. He's my husband. And we have just a ton of very talented people, everybody from biokinesiology, physical therapy, molecular biologists, imagers, anatomists, physiologists. Yeah, I work with a lot of different people, and I'm really happy to work with these teams because I certainly couldn't do it all by myself, nor am I good at all this stuff. So I get to partner with a lot of very talented people, and I get to help ask some fun questions. So I'll end it there, and if people have questions, I'm happy to take them.
Yeah. Oh, thank you so much, Dr. Petzinger. That was amazing. And I got to redo all my slides now.
Perfect.
But so incredible. We do have a couple of questions. I don't know. We did get some in an email format, but maybe people want to put them into the chat, or if you sent me that email and you want to unmute, you can ask that question. Or you could see the questions in the chat.
Chat, okay. You can now send now. Oh, sorry, that was from, I'm reading the wrong one. Chat.
Oh, okay, so here's one question. I've got one from Marco and Sue. They say, what are the options for people unable to do weight-bearing exercise to achieve these benefits? We've heard that sauna can help.
Are you saying weight bearing, like weights, carrying weights? Well, you don't have to do weight bearing necessarily. Obviously, weight bearing gives you quite a bit of intensity, right? If you do anything with weights, you're going to be, working at a higher, harder level. But you don't need weights to do that. I mean, you could also do hill climbing, and that's pretty intense. So there's a lot of ways to be intense without using weights, although weights make it more intense. You can do all sorts of stuff that would be intense. For example, even yoga can be intense, right? Holding certain poses can be pretty rigorous, right?
So, yes, sorry, they've just clarified to say not bearing weights, but not being able to stand on your feet.
Okay, well, there's other things that you can do, such as rowing, right? There's rowing, there's stationary bike, there's elliptical, so there's, different things that can do to get you working out hard. Obviously, in Parkinson's disease, as you and I both, we know, we do like to get people up regardless. And, there is a question that's come up before, and that is that, it's possible, we're exploring this a little bit, the idea that as we get older, that the cut point, what we call the level of movement to be at moderate to VIG, the cut points that we're using is aging, standard aging. But there is some data to suggest in Parkinson's disease, it might actually be a little lower than that. In other words, I might not need to work out quite so hard, but in any event, we're still exploring that. In any event, yes, there are a lot of different things you could do that aren't weight bearing, such as, do we list quite a few? I think between the group of us, we just listed a bunch.
They did want to specifically ask, and maybe you don't know about the sauna aspect. Have you heard anything about the benefits through sauna?
No, I'm sorry. I'm sticking with physical activity. Sorry, not sauna. That might be good for mindfulness, maybe. Yeah, you can't burn it off. Yeah, no, that's not going to happen. Sorry.
Okay. A question from the audience too. Eric has his hand raised. Eric, would you like to unmute and ask your question?
Yes, I understand that the different types of exercises potentially provide different benefits. I'm unclear, and could you give examples of what skilled exercise is?
That's a great question. And I apologize because I usually have a little picture of a skilled exercise. But basically, skilled exercise is anything that you're doing where you're learning a new motor movement, right? Whether that's roller skating, whether that's playing tennis, it's learning to do a type of task. Does that make sense? I'm learning to move a certain way. That could be Tai Chi. That's yoga. I'm learning to move a certain way. That's what skill is. Something that is non-skill means I'm not necessarily learning a specific movement, but I'm just more concentrating on getting my heart rate up. With that said, though, kind of to your point, it's almost impossible to think of anything. Almost anything has some level of skill. It's impossible not to have any skill, because most anything we do that's motor related will probably mean that I'm learning some type of movement, right? So it's almost impossible not to have some. But there are certain types of exercise where it's heavy skilled, where it's all about coordination and speed and coordination. Tennis, volleyball, yoga, they're very much more about getting a particular move and learning a particular task. Does that make sense?
Yes, thank you.
Okay.
Another person asked a question about when you publish a study, how do they find the results for your studies?
Great question. In PubMed.
Excellent. That's great. Thank you.
Somebody asks, will the slides be available?
Sorry, no, yeah, everything I'm showing that's out there is in papers though. So a lot of the sections I've showed that are published, particularly the first part, is all published in papers with all those figures. They're from the papers.
Right. And we are recording today, so they'll be able to rewatch the recording later.
Okay.
Jenny also has a question, right? You're on.
Okay, there I am. First of all, so I'm one of the instructors with PD Connect. Thank you so much for your research.
Absolutely.
I present it to people all the time, and I just think how you, your research is just so meaningful to clients, and I really feel like it gets people hooked to exercise, and especially the images, you know, the graphs you create. So just how you present the information is so meaningful to people. So thank you for that.
Oh, thank you.
My question to you is, usually the first follow-up question when I present your research to people is, well, should I just exercise and not take medication?
Yeah.
And I'd be curious how you answer that question.
Absolutely. Well, I answered based on data. So I didn't get a chance to show some of the data of one of my colleagues. His name is Jeff Beeler. He's in New York, at Sunny, New York. And basically he did a really nice stuff. So the short answer to that is absolutely should be optimized on meds. I mean, we don't drag people on treadmills. And some of the original work that we did on treadmills, we had patients, that had come to us not wanting to take any meds. And I can tell you out of the gate, we could never progress them on the treadmill because they really couldn't do it to begin with. So we were kind of stuck. It was really hard to progress them through a exercise program because they were too stiff and slow. More importantly, though, the data that Dr. Bailey was able to show was he took animals where they were Parkinsonian animals, and he put them through a type of motor task. And he was basically able to show exactly what we were seeing, which is the animals that were on some level of dopamine replacement and on the road and learning motor task did a lot better. I mean, their learning curve was much better than those animals that weren't. Plus, he was able to show that the physiological changes within the synapses were healthier with the animals who were optimized and on a rotarod. In fact, his argument would be if you aren't optimized, you could get maladaptive learning. And he showed in the animals that were not on dopamine and not doing well that they had evidence of maladaptive, aberrant type of connections that were not healthy. And it basically had to do with the type of glutamate receptors that shouldn't be there, that were popping up in that animal. So there is animal data absolutely that supports that statement, that you definitely want to be optimized on dopamine. And really the way that I think about dopamine, quite honestly, is this: dopamine is an enabler, right? You need dopamine because to get the most out of practice. So when you have dopamine around, it's making sure that those glutamate receptors that I showed you are being mobilized in a normal way. They sort of set it up like a chessboard so that they're a healthy chessboard, but dopamine helps that happen while the learning is occurring and while the synapses are forming. So dopamine is a very important enabler in the brain. So while it's not one without the other. Dopamine is an enabler. Again, connections aren't going to form. If I just pour dopamine on your brain, you're not going to get a synapse. Okay, there's no data for that. Dopamine is not making the synapse. Practice is making the synapse. But dopamine is making sure that when that synapse is forming, all the chess pieces are in the right place to make that connection a good connection. Does that make sense? That's a good way to say that. And animal data is very clear on that.
Thank you so much. And I'll just ask you, and you said you said so.
No, well, there's clear data that yes, you want to be optimizing dopamine. Absolutely.
Thank you.
What level were the specimens? Like level, if there are 5 levels to Parkinson's disease, were they at first level, second level, third level?
How severe? How severe were the animals? Yeah. They had 80% dopamine depletion.
Well, that's
they have. So basically, they're basically equivalent to, let's say, a moderate Parkinson's.
So what if you have early stage, and you don't have a lot of symptoms? You still suggest dopamine.
You're asking how to. Okay.
Yeah, because I'm not taking meds.
Gotcha, I understand.
So I wanted to know, at what point does that you suggest take dopamine if you're not taking it already?
Excellent point. And the truth is, there is no easy answer to that other than this. We kind of take it to the point that you're saying, we have no way. I'm actually plugging my thing in because my battery is about to die and I don't want that to die. Okay. We have no way to know where you're at with dopamine. That's true. I mean, I can't tell you where you're at, right? But what I can say is based on what you're looking like, I can tell how well you're optimized. So this is where having your movement disorder person take a look at you and say, you know what, you are looking a little slow or impaired. Does that make sense? So we just basically look, I honestly, I take what you look like. If I feel like, you know what, you're dragging, I don't want that. We want you a little better than that. Does that make sense? It sounds kind of dull and practical, but it's dull and practical. Yeah, I basically take a look at someone and say, you know what, you're slow. Let's fix that, because I want you to be able to get on the treadmill and rock it. You know what I mean? And if I see that you're dragging, I'm going, I don't want you dragging that leg on that treadmill, then I'm going to optimize, you know. And most of the time in Parkinson's, you know, the saying, start low, go slow. I mean, no one's going to hammer you with dopamine replacement. Generally speaking, they're going to start it slow and see how you feel. Then put you in some therapy, have you come back and have another discussion. So it really is I have to say, it's very practical, you know, for the reason that you just said. I can't literally put a thermometer in your brain and say, you're ready or not ready, you know. So we have to, that's where clinical experience and working with your Parkinson's specialist is really helpful. And also with your therapist. I often get feedback from the physical therapist who'll say, you know what, this person is having trouble. They can't progress. I think they're a little too stiff or slow. And I'll say, got it. Let me see them back and let's see what we can do. So it's a constant conversation. Obviously, as a patient, you're in the center of that conversation, because we want to make sure that you're feeling good while you're doing it. So you're absolutely front and center of that conversation. Your movement person is part of that conversation, and your therapist, physical therapist, is part of that conversation to help you make that decision, which, you know, again, I totally understand that there can be some gray area there, and that's where you have to work together and make a decision going, yeah, I think I should get something because I'm having some trouble here, or getting cramping or something like that. Yeah.
Thank you. Carol, will you go ahead and ask your question that you'd already put in the chat?
Sure. Can you talk a little bit, Doctor, about the relationship between anxiety and exercise? That was like one of my main symptoms when I was diagnosed with Parkinson's. And there are times when I feel it's almost become like an urge to exercise when I'm feeling anxious now.
Right. Well, you know, I tell you, everyone, I'd love to be able to tell you that we've got some data on mood. We have some hints. We're looking at that now. Most of what we've been looking at is cognition, but obviously we're very much interested in mood as well. And certainly in the regular aging field, whatever, exercise, there's lots and lots of data of exercise and depression, exercise and anxiety. So clearly seems to be some benefits there. And in Parkinson's disease, you know, there's definitely, you know, it's not usually the primary question, although there's a couple papers that have looked at that. Many times there's secondary questions that they're asking. There seems to be definitely some hints that it should help mood, you know. And again, what causes that? There's probably a couple reasons why. I mean, obviously people talk about endorphins and that sort of thing. I kind of pointed out the idea that, you know, one of the first bits of data we showed was that dopamine was being released more efficiently. And certainly dopamine itself can help with anxiety, right? When I take the pill, I feel better. I feel less anxious. So definitely, I mean, everything seems to point that exercise should be helpful for that. Now, interestingly, I have to say that for some patients, I do find that sometimes if they're working out really hard, they may start feeling a little underdosed, you know what I mean? And I sometimes feel that, okay, maybe they're not they need to be optimized a bit more. They're working out real hard and they may be pushing that envelope a little bit too much. I talk about trying to get some consistency and make sure that they're getting a more consistent effect. So we may have to optimize there. But yeah, I mean, the data for exercise and mood, though, is certainly pretty close. Yeah.
Thank you.
Excellent. Well, thank you for answering these questions. We still have a few more questions if you still have time, doctor, but if you need to go, we absolutely understand, because we know we've gone over time for you. So
yeah, I have a few more minutes. I could take a few.
Great. Excellent. Listed as Spencer.
Yeah, I'm just wondering, I just want to be really clear about what the.
Oh, no, I couldn't hear you for a second. Go ahead.
Can you hear me now?
Yeah.
I just want to be really clear what the take-home message is for exercise. Skilled exercise is more beneficial than aerobic. I mean, I always thought it was the other way around, it seemed that that's what you were saying.
Oh,
if I was going to lay out an exercise plan for myself for the week, would I have myself doing skilled exercise 3 or 4 times a week and aerobic 3 times, or?
And they're both great. We're seeing benefits with both, no question about it. So I would say both are great. What I was saying in skilled, we may see a little bit more with attention, focusing maybe with skilled, but they're both showing improvement with executive function, and they're both showing improvement with memory. So now let's think about this for a second. So I'm doing skilled, I'm doing aerobic. I can make either one of those intense, can't I? Of course I can, right? Of course I can. So if you're going to do skilled, make it intense. So I'm going to do aerobic, make it intense. If I can have a long conversation with my roommate, I'm probably not doing intense enough. I need to bring it up. And it doesn't have to be 10 minutes in a row, 15 minutes in a row. I could do 5 minutes in a row, take a little break, and then rev it up again. So
and you could do just a lot of vigorous skilled exercise. You don't have to.
Absolutely. Well, and the point is this. Why did we even talk about skill in Parkinson's? Why did we get interested in skill to begin with? Think back to the original studies. Why?
Balance.
Gait and balance. Thank you. That is why we are different than the aging field. The aging field doesn't talk skill and balance because that's not the issue as aging. It is some, but not like Parkinson's. Of course, we're always going to talk about skill, and how would we not? Anybody that has Parkinson's should be getting gait and balance practice because, like I said, you're not going to get the type of gains if all you're doing is stationary bike. I mean, unless you have a Martian brain, I don't think so. I mean, you're going to need to do skill and balance. So anybody with Parkinson's should absolutely be doing gait and balance training. I don't care what you pick. Pick Tai Chi. I could care less. As long as it's gait and balance, as long as you're making it intense. Now, is there a benefit from aerobic? Of course there is. If I get tired of skill and balance, hell, I could go out and go swimming. I could get on a stationary bike, and I could get total points for that for just aerobic practice alone, which also has benefits, but I better damn well making that intense. So the take-home message is aerobic skill always for Parkinson's, because gait and balance, I'm sorry, we need that practice. We always need that practice. And then if I'm thinking about thinking and I get tired of that, rev it up, get on a bike. But whatever you do, for God's sakes, make it moderate and vigorous, because once I start doing that, where else do I get gains? Oh, right, in my thinking. So I get double whammy. I get double points for that. I get better gait and balance, and I'm totally hitting this big guy here, the thinking, and that's what we all need. So that's why you're doing it intense. It's not just for the gait and balance. I'm driving it up so I get gains in my thinking, which is also another thing. So you're right. I could do one exercise that might hit all 4 points, right? I mean, that's the thing. That's the beauty about exercise. Many of these exercises, I can have all these different benefits for it. And that's kind of the idea too. I don't have to do just aerobic. I have to do, hell, if I play tennis, right? I'm getting lots of stuff out of that guy, right? So you can totally mix it up and do something that you love.
Yeah, great. Thank you.
On that note, people are asking about moderate to vigorous activity. Is it also more the perceived rate of exertion versus actually getting your heart rate out, or do you just want them to get out of breath?
Well, we looked at perceived. We didn't publish this yet. We're going to publish it. It's very different. It's under the moderate. I mean, so in other words, it's not matching mod vig. We got mod vig with the actigraph, and basically they were overestimating their level of intensity. So having some sort of idea of where they're at actually is helpful. Now we did not, when we asked them to tell us how hard they thought they were working, we didn't tell them that, we weren't that specific. We had some definitions, but all I can say is whenever they would write that they were exerting, many times they weren't. On the things that were obvious, like running and stuff, they were closer, but like walking, they were constantly overestimating what they were doing, and the intensity wasn't there. So I can see why sometimes having some way to gauge that is helpful. But also I would still say getting up to that level of not being able to talk is still pretty reasonable. But yeah, that is a challenge, is how hard should I be working out, and how hard I think I'm working out versus what I'm really working out at.
Right, great. All right, I think one last question, Eric, you've got your hand up there.
A lot of your research is based upon research animals. I'm just fascinated, curious, how do you get or create a Parkinson rat?
Oh, that's a whole talk in and of itself. But yeah, we have a lot of different toxin models that we can use. Scary, isn't that? But yeah, we can. The most common one is MPTP. That was discovered in the 1980s. You want to read a good book, read the book Frozen Addicts. The Frozen Addicts is a short book. It's great. It's a detective story, but basically it's the identification of a toxin that had been inadvertently developed when the chemists in the Bay Area were trying to synthesize heroin. Go figure, you synthesize heroin, you're going to get yourself in trouble. And sure enough, they had inadvertently created this toxin, MPTP. Frozen Addicts, read that. We ended up using that MPTP toxin, and we show that basically our boss in Sunnyvale, Bill Langston, showed that it could create a Parkinson's model. But there's another one called 6-hydroxydopamine. There's also something called paraquat. There's a couple of different types of toxins, but the ones we use are MPTP and 6-hydroxydopamine.
Great. Well, thank you so much. There's many messages in the chat. People love your energy. They love your enthusiasm, and you're so clear. And they're thanking you for your research, and we second that. Thank you so much for spending this
Absolutely. All right.
time
Take
with
care,
us.
guys.
Have a great day. Take care.
Thanks.
Thank you.
Bye-bye now.
Thank you.
Bye now.